In the late 19th century, a mysterious disease plagued tobacco plants. It caused the leaves to become mottled and stunted, devastating crops. Scientists were baffled. They couldn't find any bacteria or fungi responsible for the disease. This marked the beginning of our understanding of viruses, entities smaller than bacteria, invisible under ordinary microscopes. Around the same time, foot and mouth disease wreaked havoc on cattle. This highly contagious disease caused painful blisters and ulcers in the mouths and hooves of infected animals. Like the tobacco disease, no known pathogen could be identified. This further suggested the existence of an unseen infectious agent. These early observations hinted at a hidden world of microbes, too small to be seen with the technology of the time. It was a world that held both peril and promise, as we would later discover. The stage was set for a new era in biology, the study of viruses. In 1892, Dmitry Ivanovsky, a Russian biologist, made a groundbreaking discovery while investigating the tobacco disease. He found that the infectious agent could pass through a filter designed to trap bacteria. This meant the culprit was much smaller than any known bacterium. Decades later, in 1955, American biochemist Wendell Stanley achieved a major breakthrough. He successfully crystallized the tobacco mosaic virus, proving that viruses were not simply liquids or mysterious agents, but distinct particles with a definite structure. His work paved the way for understanding the physical nature of viruses. These pioneers, along with many others, unveiled the previously invisible world of viruses. Their research provided the foundation for modern virology and our ongoing quest to understand and combat these microscopic entities. Viruses are incredibly simple structures. They consist of genetic material, either DNA or RNA, encased in a protein coat called a capsid. Some viruses also have an outer envelope derived from the host cell's membrane. Unlike living cells, viruses lack the machinery to replicate on their own. To reproduce, viruses must infect living cells. They attach to specific receptors on the host cell surface, like keys fitting into locks. Once attached, they inject their genetic material into the host cell. The viral genes then hijack the host cell's machinery, forcing it to produce more viruses. This parasitic relationship between the virus and the host cell is what makes viruses so effective at spreading and causing disease. Understanding this process is crucial for developing antiviral drugs and vaccines. Viruses are most widely known for causing diseases in humans, animals, and plants. Some, like the common cold and influenza, cause relatively mild, self-limiting illnesses. Others, like HIV and Ebola, can lead to severe, life-threatening diseases. The emergence of new viruses, like the recent coronavirus outbreaks, highlights the constant threat posed by these microscopic entities. However, viruses are not always foes. Scientists have harnessed the power of viruses for beneficial purpose. In gene therapy, modified viruses can deliver therapeutic genes into cells to treat genetic diseases. Viruses are also used as vectors in vaccines, carrying harmless pieces of viral proteins to trigger an immune response. The relationship between humans and viruses is complex and constantly evolving. Continued research is essential for understanding viral pathogenesis, developing effective treatments, and harnessing the beneficial potential of these microscopic entities.